Greetings everyone, I am Srinath and today we are going to see about genetic engineering and protein modeling. First, let's see about genetic engineering. It is the process of using recombinant DNA, which is rDNA technology, to alter the genetic makeup of an organism. Traditionally, humans have manipulated genomes indirectly by controlling breeding and selecting offspring with desired traits. Genetic engineering involves direct manipulation of one-or-more genes. Most often, a gene from another species is added to an organism's genome to give it a desired phenotype. Recombinant DNA technology started with pretty simple things, cloning very small species of DNA and growing them in bacteria, and has evolved to an enormous field where whole genomes can be cloned and moved from cell to cell using variations of techniques that all would come under genetic engineering as a very broad definition. To me, genetic engineering, broadly defined, means that you're taking pieces of DNA and combining them with other pieces of DNA. This doesn't really happen in nature, but is something that you engineer in your own laboratory and test tubes, and then taking what you have engineered and propagating that in any number of different organisms that range from bags, which are bacterial cells, to yaks, which are yeast cells, to plants and animals. And before going on to next topic, let's get to another place. Here we are to the next topic, which is protein modeling and experimental protein structure determination, which go hand in hand and share the long-term aspiration of providing 3D atomic level information for most, if not all, proteins derivable from their amino acid sequences. Structural genomics is a worldwide effort focusing on the rapid determination of the substantial number of protein structures using high throughput X-ray crystallography and NMR spectroscopy. Populating the PDB with determined structures and therefore homology templates corresponding to major protein families is essential to first limit and eventually eliminate homology, which is no-go areas within proteomes, so that most and eventually all of the remaining sequences can be accurately modeled. These new structures will also enrich the de novo methods that sample the PDB. Structural genomics initiatives have contributed the majority of new structurally characterized families and over 5 times as many novel folds as classical structural biology. Most structural genomics consortia apply bioinformatics strategies to selectively target specific structure, function in particular class of proteins such as membrane proteins, clinically important protein families, proteins containing specific domains, and in determining all the proteins in a model genome. However, even with the structural genomics, burgeoning growth in genome sequencing data means that structural inferences will be made for the vast majority of proteins on the basis of structural modeling rather than experimental elucidations. The continued careful selection of structural genomics targets is therefore essential and should specifically aim to ensure that majority of sequences that cannot currently be modeled are related to at least one experimentally determined structure of 30% sequence identity or above. It has been estimated that a minimum of 16,000 targets must be determined to cover 90% of all protein domain families, including those of membrane proteins at 30% identity or maybe above. A further challenge is presented by metagenomic projects based on shotgun sequencing of populations of microbes, which have suggested a linear relationship between number of protein sequence and the number of sequence clusters, and therefore families, and that there are likely many proteins families that remain to be discovered. In preparation for the emergence of improved homolog sets and challenge of new protein families, case for continued development of combined homology modeling or de novo modeling approaches is reinforced. With this, a miniature 3D model of protein is obtained. In a nutshell, genetic engineering and protein modeling are like the best friends. When they join together, it is like bringing dry lab and wet lab on the same pace. With protein modeling, process consumes less time and is a precursor for genetic engineering works, whereas the rest is done by genetic lab in real time. Thank you for listening.